Hello, welcome to the IBH seminar today. I'm Micha. I'm here with my colleagues Craig Simpson. They're out of view because we're just sharing from one laptop, but they're both in the room. I've got Milne here as well. So Linda and Craig and myself have been working on this project together with two other colleagues for the last few years. It's a barley gene expression database called EORNA. And today what we want to talk you through is a major update to that database. And we'll be showing you some nice examples of use cases as well. So we've split it into three parts, our talk. So initially I'll be giving you an introduction to EORNA itself, a summary of the new features, the automation of our workflows, and the second version of EORNA. And then I'll hand over to my colleague Craig, who will be talking you through the new features and give you some nice examples of biological use cases. And then at the end, we have a question and answer session between all three of us. And yeah, so we'll take it from there. So we started working on this roughly in 2018. And really, EORNA was born out of the observation that an awful lot of RNA-seq was accumulating in public read archives. And growth then was exponential, and it's still exponential. And you can see from the graph on the right-hand side that we've now got about 90 petabytes of data in the European Nucleotide Archive. So this data is all subject to FAIR principles, which means it is meant to be reusable. But we're actually seeing very little evidence that people are reusing it and reanalyzing it. And we've repeatedly made the point that people have started using research data like single-use plastic, and we don't think that's good. So with that in mind, we set about developing EORNA, the first version of EORNA. So EORNA is a gene expression database for barley, and it is based on quantifying the data from the European Nucleotide Archive. So all the public RNA-seq in there. And our first version of this was published in 2021. And back then, it was a fairly modest proposition compared to what we have now. So it was 843 samples from 22 studies. And it has been quite popular. We've just got new user stats this week. And we've had 660,000 hits from 75,000 discrete IP addresses since January 2019. So people have clearly been using it, and that's a great thing. So when we came to the end of the work on EORNA 1, we realized that because of the exponential growth of the data, it was going to be impossible to make any kind of future updates to this database without automating an awful lot of the data handling, which at the time was mainly manual. And with that in mind, we went to the BWIS-RC and applied for funding for a major update to EORNA. So this was a grant that ran from 23 to 25. We've just finished work on it. And it revolved around three major work packages. So the first work package was all about fully automating the whole process of discovering the data, retrieving the data, and quantifying it. The second work package was about producing a new generation of EORNA database, this time using all the publicly available RNA-seq data, and also using the latest reference transcriptome. And then the third work package was about open science, really, and just taking the whole infrastructure that we had created and making that available freely to the community so that people can make their own EORNA databases for other species. So that included our data processing pipeline, the database schema, the web front end, and also the quantification data from this project. So on the topic of automation, so we've been using the API at the European Nucleotide Archive for this. So an API is an application programming interface, and it's basically a recipe for making software programs communicate with each other directly. So we write a bit of client code and it talks to the API at the ENA. So the first stage in this is then to send a query to the API that discovers all the studies that contain barley RNA-seq data. And then you iterate over that list, and for each of the studies, you post another query that returns a list of all the sequencing runs that belong to that project. And then you can iterate over these. They each come with a URL where you download the raw reads. You then quality trim them, and then you quantify the expression using Salmon. So this is all completely unsupervised and doesn't require any human interaction. And it scales beautifully because of that. So initially we implemented that just as simple shell scripts, but then latterly we have been turning these into proper production quality workflows. So we're using a system for this that's called Nextflow. And it's a very modern, very powerful workflow management system. One of the beauties is you can take someone else's workflow and you can deploy it on any kind of scheduling system, so any kind of high-performance compute cluster. And you can even put it on Amazon Web Services. It handles the deployment of any software that the workflow needs for you, so you don't need to worry about that. And Nextflow itself is very easy to install. So this is a great system and we're really happy with it. And what this means is we can give this to other people and they can have their own EORNA databases. So we also made a new reference transcript dataset, or RTD, for EORNA v2. So the main part of this was the pan transcriptome, which was just recently published. So the pan transcriptome is based on 20 genotypes from the first barley pan genome, and you can see a list on the right-hand side here. And each of the genotypes is represented by 5 different tissues and 3 bioreps. And then we combined this with 2 other existing RTDs. So first, the previous reference transcriptome from cultivar Barker, which is called BART version 2. And then the Morix Atlas RTD, which was published alongside the pan transcriptome. And that contributes additional stress genes for biotic and abiotic stresses, which weren't well represented in the other 2 RTDs. And between these, between the 3 of these, we've now got a very comprehensive set of transcripts. And that was important because we wanted to have as big a mapping space for our data as possible. So EORNA2 RTD has just over 87,000 genes, just over 650,000 transcripts. That may sound like a lot, but we're actually seeing that pretty much all of these have expression against them, and less than 0.5% show no expression in our data set. So the current status then of EORNA v2 is that we've got 171 study accessions. We've got 6,285 samples from these studies, and they're all vulgar and spontaneum. It was just over 12 terabytes of raw data that we had to process for this, and it took a total of 6 CPU years to process that data. But we got it done in 3 months of walk clock time. So the biological scope of the database is really quite broad. We've got 921 different varieties. We've got 144 different tissues. We've got 231 different developmental stages. And the whole thing is now live at the URL that you can see at the bottom of the slide. But Craig later will talk you through the details of how you use it. And I've got one more slide before I hand over to Craig. So we should really be talking about the challenges that we had as well. And the main challenge by far was the metadata. generally what we found was that there is too little. People don't put in enough metadata. But often also we found that fields in the submission template get used inappropriately. So for example, people will put cultivar names in a description field rather than the cultivar field. We found inconsistencies in how things are spelled or described, and the list just goes on. And the bottom line from all this was that we had to do quite a lot of manual curation. And when I say we, it was mainly Craig and Milne who actually did this. And we think going forward, there's probably space for an AI-based solution that can deal with these problems automatically. But I would also like to issue an appeal to the community to take time and make effort in configuring your metadata carefully before you submit them, because it just helps them having that second life that they should be having. So with this, I'm going now to hand over to my colleague Craig, who's right next to me.
Hi.
And he will talk you through how to use the new version of EORNA and give you some nice biological use cases.
Okay, thanks, Micha. So I'm the biologist and I'm the user of EORNA2, and I hope some of you will be able to use it quite frequently as well. So what I plan to do is to go through the website and the database, how to find your gene of interest, and then the information that is offered by the database. And I'm also going to give you some pointers on how that information can help you in answering different questions. And you never know, it may help you support your grant applications and your papers. So the great thing about EORNA2 is that it gives you really strong insights into barley biology and uncovers fundamental gene and transcript responses to barley growth, development, and also to stress responses. So I think the information that it holds will be useful to you, even if you're not that interested in gene expression. As Micha's already said, it gives you the information on 171 different barley gene expression studies, and it links them to the genomic information. So, as I've said, EORNA generates questions and hypotheses, and also helps answer some questions about your genes of interest. So at the very end, if we have time and if people want me to, I can do a live version run through of the website, and hopefully I'll be able to deal with any issues or comments or ideas that you may have. Okay, so let's go. So the first thing to say is why are we doing it? Well, it's very important. So things like plants are, you can have genetically identical plants that can produce very different plants under very different environments or growth management practices. So the foundation for these changes are in the changes in programs of gene expression. So if we can understand gene expression, we can then understand how a plant responds to growth development and its environment. So gene expression is important, and what EORNA2 does is it reports those gene and transcript expression levels. So this is the front page of the database. So you can see at the kind of top, there's a little bit of an introductory part. And then this is followed by a group of 6 boxes. So, and this is how you use the database to find the gene of interest that you have. So I think we all know how to use BLAST research, but there is also boxes. So there's a box here, annotation search. So you can put in things like general annotation information, for example, say auxin, and you'll then find a list of genes that are kind of linked to auxin. As we know, barley, one of the great things about barley is we have many kind of different gene names for barley genes. So that what this does is translate a list of all the aliases of gene barley names that we have, and you can translate that, and you can then find your EORNA2 gene that's there. I'll describe a little later about Go enrichment. These two, the search by idea and the region search, these are kind of linked to us utilizing the barley 50K SNP data set and how that maps to the genome, or different parts of the genome that you can find, and then basically the EORNA2 genes that are associated with these regions. So these are the kind of 6 main ways in which we can then go in and try and find our gene. I'll come down to the filter studies in just a little while. So the main output from EORNA2 are these graphs that we get. So the graphs are basically showing measurements of the TPM values on the y-axis here. So this is TPM values, and then along the bottom here is every single sample. So in this case, there's 6,285 samples. For each sample, there is a little tag, and the little tag will give you some metadata information on each of the samples if you want to go into that. And you can then have the slide bar, and you can slide it along, and you will then find all 6,285 samples. What we also are able to offer is an Excel file sheet with all the TPM values. So you can just click on that there, and you can then download all that information to your own computer and do the analysis that you want. You can adapt what you look at by looking at the study details. So there's 171 different studies with 6,000 different samples. You can load that up, and you can then be more selective in what you look at instead of looking at everything. And you can go in individual detail, look at individual runs as well. You can pick out individual runs and say, I want to look at this and this, and then pick those out. And then you can then generate the graphs specifically for those things. So these 3 bits here are extremely useful for kind of focusing in on the information that you need for your gene. So I'm going to make a little comment here about plotting. We spent quite a bit of time trying to improve the efficiency of plotting, and we're using different graphics now, which makes it much quicker. And it does work generally it works very quickly. But if you've got a gene, say, that's got 500 transcripts on it, and you're trying to look across 6,285 samples, it will take time. So you just have to be patient. And if it's taken, if it is something that's got a lot of transcripts in it, maybe you want to go and have a coffee, get the coffee sorted out, and then come back to the information when you're ready. So I just want to kind of make that clear at the start. Don't sit there going, why is this not coming up? Everything is plotted real time on your computer. So the one great thing about the examples of genes that we can find very quickly, and that's very intuitive to look at, are examples of genes that are very tissue and condition specific. So I've given you a couple of examples here. The first example is gibberellin 2 oxidase that was described in the pan-transcriptome paper that we did. And we find that that has very exclusive expression in developing seed and grain. And you can see that quite clearly in all 6,000 samples across, that where we are seeing the expression is things that are related to grain. Similarly, examples where we're looking at abiotic stresses, for example. So in this case, I use a well-known cold response gene called 14B, which is very exclusively expressed in response to low temperature. And again, you can pick out all the different examples of genes that are responding to the cold. So using EORNA2 in this way, using the multiple different and separate experiments, gives you a real confidence you have a true tissue condition specific expression, and it's really valuable on its own just for that. So the main thing with 171 RNA-seq samples studies is that it is quite difficult to plot in a visually pleasing way. So this is one example, and this is the sort of data you can get, and it's not that easy to use. So one of the new things that we've done in EORNA2 is to develop a filtering system so that we can filter by different experiments, by different tissues, by different cultivars, development stages, etc. And you can see that on the front page in this region here. So in this region here, you've got your gene name, and then you could add in study keywords. So the study keywords is a part of the table that we have added in, and it's based on the study related to the publication that was given for each of the study accession. And we can then also add in cultivars, which is in this part of the table. There's also tissues, there's developmental stages, and also some of the experimental conditions. and also replications. And they can all be put in this part here that allows you then to search. You click on there and you can then go searching for the matching studies that's related to that. An alternative way to do it is to use this box here. You can then click on a number of different studies that you want to look at, and then you can go and search for that. On the front page, we've given you the whole table of all. If you can go all the way down the bottom of the page, you'll see all of the 171 different studies, and you can get the information that you want from there. So filtering has been really helpful. So after you've gone through the filtering process, or if you want to have all 171, you'll be moved on to the next level. And what you have is this page here. And this page here basically has all the studies, and you then basically select what studies you want to look at by just clicking on here. If you don't click on anything, you'll get all 171 studies. So that's how we then get into the graphs and the information that we're after. So I find using EORNA2 is really very useful, and one of its real nice uses is to try and find what is the most likely protein coding sequence. So transcription and RNA processing can be a really messy business. Most of the RNA extractions that we do involves having the nucleus and the cytoplasm there. And the nucleus is known to be quite filled up with a lot of partially processed messages, which then get incorporated into the RTD that we use to quantify against. So I've given you this example here, which is a U2F35A, it's a splicing factor, and it has 17 different transcripts. But you can see from the information, and I've just taken one study only, which is 16 developmental tissues, you can see that there are kind of 3 main transcripts there. So out of all the 17 transcripts, there are 3 main transcripts that seem to be transcribed at any sort of level of expression. So you can then pull out and find out what these transcripts are. So in this case, we've got 3 transcripts, which is 8, 10, and 12. And you can see the gene models for those here on the right-hand side. So if you take those transcripts, you then translate them. You can then generate the reading frames over 3 reading frames. You can look at the data and you find very quickly that your expected translation start is here. And you find that in transcripts 8 and 10, there are upstream open reading frames. So upstream open reading frames are really important translational cis regulatory elements. My friends here, Martin Balcerowicz and Emma Hardy, they would describe them as speed bumps. So basically they slow down the translation of the main open reading frame, or they bump the transcript off the ribosome. So in other words, what upstream open reading frames do is to make translation less efficient in those transcripts. So you can see here that transcript 12 doesn't have any of those upstream open reading frames, and it's got a full length coding sequence. So transcript 12 is the transcript I would see as being the most important transcript in terms of translation. So a nice thing you can do with the graphs that we have is you can then click off every single transcript that you don't want to see. So in this case, I've clicked them all off except for transcript 12, and you can then see the expression for what I think is the important coding sequence transcript. And if you compare that then with what was at the start, you can see that there was mostly transcript 10 and things like the epidermis or etiolated leaf, and which there is very little of transcript 12 in those different tissues. So you can see that you can very quickly generate questions, and you can very quickly generate a hypothesis about why that might be and what sort of effects this may have on the expression of your gene of interest. Okay, back to the database. So we use JBrowser to link the RTB, our reference transcript data set transcripts to the expression data to the genome. And the genome we are using is the pan genome 20 that was generated for the pan genotypes. So this is to allow us to see all the different transcripts that we might have within the reference transcript data set. So the nice thing that we can do is you can select for all the different gene aliases, and you can then see all those gene models. So you can find the ORNA2 gene models, and you can then line them up with all the different other gene aliases that we have. So, and then this is, like Morex or the Horvu gene 1 and 3, also the original BART version 1 transcripts, some people remember, they might remember MLOX and JLOX. So they've been around for many years. So all these can be selected, and the gene model will be aligned up in the browser. Again, the nice thing with the JBrowse is that you can click on the individual gene models, and it will generate this kind of different feature details that are related to that gene model. And you can pick out then the coding sequence, protein sequence, cDNA, etc., etc. So we also supply the individual sequences. So if you go down slightly further down that page below the browser, you'll find that you can then click on every single individual transcript. We can also open up every single potential coding sequence. We don't have the upstream open reading frame, so you have to go and work that out yourselves. But that information is there for you to utilize if you want to. Down here, we have all the different gene aliases related to that ORNA2 gene, which again, you can click on and move to different sites that allow you to look at those genes in a bit more detail. Further down, we also have the best homologies to the orthologs for rice, for Arabidopsis, and also Brachydipodium. So it gives you some sort of idea of what your gene is doing, and it kind of helps with the annotation. And we've also looked at the functional annotation for those individual genes, and we've kind of linked that up to TRAPID, AHRD, and also GoIDs by Panzer. So all this information is there and available to you. Okay, so now I'm going to move over to that block called Go Annotation. So again, we've generated a page that allows you to try and link your gene to some of the annotations that are linked to that gene, and also to kind of give you some of the Go tags that are associated with those genes. So here we have, first of all, we have this Excel sheet that you can download, and you can then, if you do that, you'll have a page something like this here, and you can add in a list of different genes, ORNA2 genes, and then it will tell you all the different Go annotations associated with that. So here's just kind of an example of a few genes here. This gives basically the Interpro TRAPID identifier tags, and it'll give you some annotation. So this will give you some idea of what sort of gene that you're working with. The other thing that's available to you is to look at Go enrichment. So we use gProfiler to do that. And so to do that, you need a GMT file. So the GMT file is basically linking the gene name to the Go tags. So you add those to basically click on the custom GAT. You download this file down the very bottom. I hope everyone can see that. Just right down the very bottom. You can download that file. You can load it up into the custom GMT, and then you can add in your list of different genes that you're interested in, and it will then give you an enrichment, give you some idea of the genes and what annotation of Go tags are kind of linked to. So that can all be done for you as well. Okay, so I'm going to move slightly differently now. So the thing about ORNA2 is everything about ORNA2 is massive. It's big. We're talking about 80-odd thousand genes. We're talking a whole pile of different tissues and conditions. And in addition to that, it's extremely rich. There's over 850,000 transcripts that it contains within the reference transcript data set. So these variants are the results of, are the result of different genotype-specific differences and transcriptional and post-transcriptional processes such as alternative splicing and also alternative transcript starts and also transcript ends. So I'm going to give you a couple of examples of genotype-specific transcripts and alternatively spliced transcripts. That doesn't mean to say this transcript starts and ends are not important, but a lot of the things that the consequences of these is very similar to some of the things that you'll find in alternative splicing. So first of all, the genotype transcript variants. So these ones, so this is, so what is really really nice about EORNA2 is it has a whole pile of different diversity panels, which we didn't have before on EORNA version 1. So that allows you to study in quite detail, quite a lot of detail, the different genotype differences in gene expression. So the example I've given here is one tissue from 209 genotypes that were registered for the UK market. And this was from Miriam Schreiber's paper in 2024. So if you look across the different genotypes, you can see that there's two colors. There's a dark blue and a light blue. So if you look at, find out what those two are, you find that there are two different transcripts, basically 6 and 10. So we can look at that in more detail across other genotypes. And so the example I've given here is a set sample, which is Clipper and Sahara. So you can see again, it's moved from this kind of light blue transcript 6 to the darker blue transcript 10 in Sahara. If you look across Morex and Bowman, both of those are following transcript 6. But then if you go into other data sets, you'll find other genotypes, and they have a completely different type of transcript there, which is in this WI4330. So the nice thing is you can link up with some of the genomic data, and you can also identify some of this variation and how it lines up. So in the case of this, you've basically got four different amounts of 4 base pair repeats that have been deleted. So in the case of Sahara, there's a 4 base pair deletion compared to Clipper, and in the WI4330, there's an 8 base pair deletion compared to Clipper. So you can go and characterize these different transcript variants very quickly. So alternative splicing. So I would say that differential gene expression is not enough. So gene transcription may remain constant, but what we then miss is what is underlying the changes in transcription. So one of the really nice unique selling points of EORNA2 is it's looking at individual transcripts and how those expression levels change. And so you can use EORNA2 to kind of get a much better idea of what's going on at alternative splicing level compared to if you were looking at differential gene expression. So example I've given is from a paper in 2018, which is basically showing that there's a fairly general constant level of transcription going on, but underlying that there are 2 transcripts and they are switching between normal temperature to low temperature. So there is a switch that's gone on here. So basically what I'm saying here is, well, we need to start spending a little bit more time looking at the different transcript levels, and that they are important. And that EORNA2 allows us to look at those different levels of transcript expression. So the example I've given you here of a switch like this is a gene called Gigantia. So Gigantia plays a role in the cold stress response, and specifically in freezing tolerance, and it influences flowering time. It's induced by cold stress, and mutants of the GI gene are very sensitive to freezing. The other thing to say is that it's independent of the important CBF response pathway. So I've used 2 data sets here. One is our 16 data set, 16 developmental tissues, and you can see that it's staying with this kind of green transcript pretty much all the way through. This is because temperature is not affecting it in any great way. However, if you another data set that I used was an overexpression of the wheat CBF gene and different CBF genes. And then they've been tested against normal temperature and cold temperature. And you can see that under cold temperature, you get an induction of this darker blue transcript. So again, we can go and look at those different transcripts. And we find, again, we're back into our 5' UTRs, and we're also finding that an exon skip that's going on in the cold. And this then reduces the size of that, and you then lose an upstream open reading frame that you find in the green transcript. There is another transcript there, which is this purple transcript here. So this, again, is a gene that basically is losing its N-terminal sequence. So I haven't really shown you that information. The point here is that you can see quite distinct switches going on that are the result of the change in alternate splicing in response to the condition. And EORNA2 gives you that information. Okay, finally, how am I doing for time? All right, so finally, I want to kind of make you aware of the effect of using TPM values. So TPMs are currently the best way of normalizing RNA-seq data that allows us to compare between samples. So normalization is to 1 million TPM total across all the genes for each sample. So if you were to take one sample and add up every single TPM at the very bottom, when you've added them all up, you will have 1 million TPMs. And these TPM values come directly from outputs of salmon, and then in EORNA2, that's what we plot. So basically all we're doing here is giving you the raw data. We're not doing anything fancy with that data. We're not doing any statistical analysis on it. So we have no consideration for the batch differences between samples. We don't have any kind of differences among experimental studies. We're not looking at scale, and we're not looking at statistical significances. So just be aware that what we're giving you is the raw data that allows you to go and do your analysis of interest. However, I'm going to give you, so the idea that we have here is that we generate some intuitive rapid visual assessment of the transcript expression levels of barley genes. But that is not always the case. And what I want you to make you aware of things like photosynthetic versus non-photosynthetic genes. So Rubisco activase is a well-known photosynthetic gene, and you can see that it generates a lot of TPM values when it's in the photosynthetic tissue and not in the non-photosynthetic tissue. So the issue for that in terms of the TPM values is that there are 3 genes, RBCS1A genes, and Rubisco activase use 8.5% of the TPM values. And that if you look further afield, there's 95 genes in shoot that use 40% of the TPM values compared to the same genes in roots. So the effect on that is there's less TPMs available for some of the other genes. So I've given you 2 examples here, U1A and U1C. Again, they're splicing factor genes. We'd expect those genes to be fairly constantly expressed across all living growing tissue. And you can see that in the shoots, it's much lower in levels. So the effect of that is, one, it's either due to there is a reduction in the amount of expression within the shoots, or it's due to there's less TPMs available for the quantification. So this is something just to be aware of. You can deal with it in different ways. One is to use housekeeping gene normalizations, and the other is to maybe use median normalization. So you could use these to kind of generate data that is much more consistent across all the samples that you're looking at. But again, we are not doing that here with EORNA2. We are just giving you the raw data that allows you to do this analysis. And what I'm trying to make you aware of is things like photosynthetic tissue versus non-photosynthetic tissue can lead to changes in the amounts of TPMs that you see. So, I mean, I would kind of finally say that you need to use the values that EORNA gives you to perform your own types of analysis. So we can then, even if it's photosynthetic or non-photosynthetic, you'll still see examples of genes that are significantly changing in fold differences and you can do the analysis to work those out. Another nice thing that potentially could happen in the future is that we can the identification of these genes and how they link to the genomes will allow you to link some of the genotype markers to gene expression and the complementary Manhattan plots that you may have. And we have in EORNA2 mapped in all the 50K SNP chip for barley, and they've been mapped to the genome. So you'll be able to kind of link the expression values and levels that you have for individual genes to them. So just to say, expression data is ready at your fingertips. The EORNA2 RTD is very rich in transcript variants. And thirdly, there's such a broad range of tissues, organs, developmental stages, and stress responses to choose from that you can go and look at and generate your own questions that, and hopefully some of the answers will also come from the data that's presented. So just to finish with thank you. So there's a whole pile of people that have been involved with setting up the idea of EORNA database from the very beginning, and these are kind of named on the left. And then we've also had support and funding from BBSLC and the Scottish Government. So I will stop there.
Just going to stop sharing so we can actually see the chat, etc. We weren't able to see the chat. I hope there wasn't anything in there in between. So there's questions. Do you want to come around? We'll try and huddle around the screen. We've only got one laptop here to share. We'll try and huddle around it for the question and answer session.
Micha, there's only one question in the chat just now. I don't know if can you see the chat?
Yeah, I can see a question from Flavia. Is that the only and first one? Yeah. Okay, so the question is, given that TPM is a within sample normalization salmon, how should users interpret TPM differences across independent experiments in EORNA2?
So I can give a fairly quick answer to that, is that TPMs are designed, so they're different from FPKM values. So TPMs are designed to allow you to compare between samples. Is basically the simple answer to that.
Yeah, so everything is mapped to the same RTD, and therefore, in theory, things are directly comparable within samples and between samples. That's the idea with TPMs.
There's a hand up.
Okay.
I'll
says my question was before the perfect slides that you have addressed, so that's fine. So it's all good.
I'll just unmute Run Swang and he can ask a question.
Yeah.
Hello, hi.
Thank
you for the great. Can you hear me okay?
Yes.
Yeah.
Thanks for the great presentation. I just have a question, probably not directly related to the EORNA2, but I was just wondering, what is the stage of the pan-genome graph development now? What's the possibility to map those transcripts to visualize on the pan-genome graph? Is that like reality in the near future, or?
It's not reality right now. We still have the scalability issues with the pan-genome graphs that we had 2, 3 years ago. We're not at the point where we can routinely use graphs as a drop-in replacement for linear reference sequences, unfortunately. And I think it's very hard to put a figure on at what point in the future that is actually going to happen. I mean, there's now an increasing awareness among the developers of graph tools that at the moment in the plant community we're not well served by what is out there.
Okay.
And there's attempts to change that, but how that is going to link to using graphs for transcript mapping, I really can't say at this point.
Okay.
But
that's all right. Thank you. I just want to update. Want to keep up with what's going on out there.
Yeah,
thank you.
I'll make sure you'll be the first person to know when we successfully map transcripts to a graph.
All right, thanks.
So Davida, I think.
Davida's.
Davida had his hand up.
So, hi there, can you hear me?
Yes.
Yeah, we can hear you, Davida.
Okay, so it's a recurrent question, guys, that I ask you privately many times. We've got the gene that is present in bark and absent in Morex. We have the BART-V2 ID, we double check it, but when we try to go into EORNA2, we can't find the corresponding, let's say, the corresponding data. And we know we have provided the raw data for building up EORNA. I wonder whether this has to do with the, let's say, polymorphism, or there is anything else that we should do to mine the database in this particular case or for similar cases.
That's an odd one because in theory, bark is in there. Bark is represented really well in the RTD because we included BART-V2 for backward compatibility for exactly these kind of reasons. And bark is also Barker was also one of the genotypes in the pan transcriptome. So it should be in, it should be really well represented in there. And I don't I can't think of any reason why you wouldn't be able to find your gene there.
Can I,
Perhaps, yeah.
can I ask, when did you do a search for the,
5 minutes ago, during the presentation?
okay.
Good, good.
Well, you may need to come and speak to us about this separately because that really requires investigation. But Barker should be very well represented in there. So there may be something else going on that we're not aware of.
Okay.
Can you kind of look at BART version 2 on its
Yeah,
own?
no, I can re-find it. So if
Can you?
if I let's say blast the sequences and I look into BART V2, I actually get back the ID that should be, let's say, the corresponding one. So I have no problem with BART V2. Is the other let's say is the other data set that can find the gene.
So is this the gene that Tasnim's kind of?
Yeah, we are working on the same things. Is the same gene.
Right.
Right. We need to
We'll
look
have
at
a
that.
look.
You can send us an email and we will have a look at it. Runshan is saying it could be tissue or condition specific.
Yeah, it could. I mean, we have identified it in roots with our own RNA-seq, and we map back to the BART V2. But yeah, I mean, happy to explore further. Let's say if there is an easy fix, that would be great. But if you need to explore more, yeah, we can keep going with the discussion.
If it's in BART V2, then technically it should be in EORNA V2 because BART V2 is meant to be a subset of EORNA V2, unless something has dropped out because of filtering that Wenbin might have applied. So this was the RTD was put together by our colleague Wenbin Guo, who sadly isn't here today to answer questions about it, but we could have a look at whether he applied any filtering that might have, where we might have lost things. I really hope we haven't, but you can never rule things like that out. So,
It could be a tissue specific thing. So I can't remember the BART2 using infection samples. So it may be that the infection is not there. So it might be a very specifically expressed gene in under infected tissue.
Okay, but I'm very happy if if you want to bring the case further to keep discussing.
yeah.
Yep, we can do that. There's a Martin Barcelovic.
Yep. Hope you can hear and see me.
Yeah.
Really useful resource, obviously. I have a very technical question, I guess, with all the different transcript versions that you identify. How stringent are you, and what do you define as a different transcript? If we went to the extreme that is a transcript with one additional base pair at the transcriptional starter inside a different transcript version, etc. Could you comment on that?
Do you want to answer that? So there won't be SNPs. So a transcript that's got one nucleotide difference will not come up as two different transcripts. But the example I gave you is one where there's like 4 base pair indels that are there. So I was looking at ones that were quite a low level. You can obviously see transcripts that have got bigger indels. So I haven't seen anything below the 4 base pair level, but they may be there. I haven't actually looked at them, but I think I'm pretty certain there won't be transcripts that are differ by a single nucleotide.
Yeah, I was also interested whether you would pool something that has a 1 or 2 base pair shift at the transcriptional start site. Is that then considered one transcripts?
So there are different transcriptional start sites there, and some of them are quite short. But we used the BART version 2, and so that was one way in which we tried to tidy up the different transcripts at the transcriptional start, because we're just getting so many transcripts that were varying at the 5' and 3' ends. So statistically, Ranjwan will be able to answer this better than me. So we were able to kind of reduce the number of transcripts that are to a kind of more statistically relevant level of transcript starts and ends. Ranjwan, you'll be able to answer this better than me.
I can comment a little bit on that. So so the transcript start and end sites are only determined by the long reads, because the short reads you're not really sure where it starts, where it ends. So the transcript start and end are not determined by the short reads.
Right.
The long reads are used to determine the start and ends, and we have to see significant support going. That has to go through a kind of a probability test, and any difference between 50 nucleotides will be merged together. So you probably won't see any like alternative transcript starter and less than 50 nucleotides. If it occurs, let us know.
Brilliant. Thank you so much, Ranjwan. That was exactly what I was looking for.
Yeah.
Just to follow that up also, because most of the transcripts actually come from the pan-transcriptome. The pan-transcriptome paper in Nature Genetics from last year is a good place to look at more detail of how the RTDs have actually been put together, and it's all following that kind of approach. We've inherited most of the transcripts in EORNA v2 from the pan-transcriptome directly.
Great, thank you.
Any more questions? Nope.
There doesn't seem to be any new ones in the chat.
Okay.
Any more? No one?
Would you like me to have a live session with Okay, so how do we get this live?
How we're doing for time, Malcolm? Is there time to get one thumbs up from Laura?
The webinar is scheduled for another half an hour, so yeah.
Well, probably won't be half an hour, but it'll give you kind of a flavor of the website and how you can use it. Okay, so is this screen up on?
Yeah, you need to go back to sharing.
Yeah,
Hold it. It's weird because that second window just disappears. Malcolm, can you shout if you can see the web browser?
Yep, looks good.
Yep. Okay.
So I'm just going to give you a quick example. So I've basically pasted in a bit of protein sequence from a gene, and then we can kind of go through the use of the EORNA2 website, see where we get to. So the first thing it says that it's a protein, so we need to select that as a protein. The e-value cutoffs, I generally leave it at this sort of level. Obviously, you can become more stringent or you can be less stringent as you want to do, but generally this sort of level gives you a good number of matches coming up that you want to do. So I'm going to look at ORNA2, but you can pull out the different sequences related to all these different RTDs and different assemblies if you want to, but I'm just going to focus in on ORNA2, obviously. So I can then blast that sequence. So what it generates is really, I think, quite valuable in itself. So again, here is showing the different transcripts for this gene here, which matches the best with your gene. So this is a good thing. But if you look further down, there are also other matches. So there's a good match with the 5' end or the N-terminal sequence of the peptide. There's good matches there at this side. And if I was to go and look at that, I would find another gene, which is probably an ortholog, or sorry, paralog of the gene of interest. So this information here is actually quite valuable. And again, you can click on that and you'll go straight to the selection process of the thing. So you can filter the studies if you want to, or you can set, so I'm just going to set like the 16 data set and see what comes up, just out of simplicity. So basically it's come up with the one. So I started off with 171 samples and now I'm just down to the one, and I can then go ahead and do that. So if I go back just to kind of, oh, actually I'll do that. So then you can click on that. If you had like 10 different studies, you can select the sort of studies you want from that, or you can look at all 10, for example. And then you just view the TPM values. There you go. So it's taken a little bit of time to plot, but hopefully it won't take too long. And you'll see this is one of the examples I used previously. So this generates the information across all the samples for these 16 different barley developmental stages. And you see the different levels of the TPM values going along, and these are all the different samples. And again, you get all the different metadata tags associated with them. I mentioned before that you can reduce. Remember I said that 12 was kind of an important data set. If you click on that, you then lose that information. So that data then becomes available to you. So you can play around with that. If you click on the top right here, you can either print it off or you can save the image as a JPEG or a PNG file. So that information's there for you as well. If I do that, you can then download the Excel sheet, and this has all the different experimental information, and it has all the sample data, the tissue type, developmental stage, and then all the TPM values across. So you can then utilize those numbers to do your own different analysis, if you like. The table, so if I click on that, you can see the table I've picked up. It's a bit unfortunate. I've only kind of brought up one table, but that it would, if you had 10 tables, it would come up with a 10, and you can then do further selection if you want to specifically pick up on the one thing, and then redraw then you can redraw the graph. And within the experiment, you can pick out all the different individual sample sets. So if you only wanted to look at the Caryopsis here, I'll just go down a bit more. So if you want just to look at Caryopsis and not look at any of the other things, you can then kind of click on all those. So it takes a wee bit of time to click. I'll just do it very quickly. Should use the mousing. So then you can redraw the graph. It's still running. Didn't take too long. So now I basically clicked on 6 of the samples, and you generate the information for those 6 samples. This is the browser it's telling you about. Again, you can, if you go up here, you can select if you want to, for example, the BART2 genes, you can select those and they will then come up. Gently reduce the size of this. So you can then pick out all the individual BART gene models and match them up with the ORNA2 gene models that you've got. So that's all available to you. You can adapt up here. You can do various things with the browser. So, you know, you can change the colors. You can show different things, like we've got all these kind of vertical lines. You can take those away. So you can adapt the way that the browser looks if that's what interests you. Okay. As I said, you can open up all individual sequences for every individual transcript. So those 17 transcripts. And then you can open up the individual protein sequences of you that are predicted from those individual transcripts. In this case here, all these transcripts are expected to produce the main coding sequence. Here is the individual aliases of the different genes. So as I said, you can click on these and you can end up going back to different experiments from yesteryear, if you want. And then you've also obviously got the data that's kind of linked to orthologs and different plant species. And then these are the different tags. So that gives you kind of a flavor for the different things you can do.
The graph is similar. So
Oh, okay. So yes. So the thing that Linda's just mentioned. So you can zoom in on different parts of the graph if you want to get a kind of more clearer picture. You can go back. Okay, that's it. Any further questions? That'll be no.
API available. Excuse me.
well,
API.
in a way, you can probably manipulate the URLs, so everything on the search is visible in the URL. So you could potentially just save a URL for your favorite gene, rather than having to go back through the process of what you just did. I think that would probably be the equivalent of that.
Any more questions from anyone? Laura?
Laura. Yeah. Do you want to give
Malcolm, I think you need to unmute Laura.
Hi, thank you so
Hi,
much.
Laura.
Ciao, nice to see you. Thank you so much. This looks brilliant. Just a question. If we have new study, new data sets, is it still possible to submit them to you guys to integrate in the database, or I mean, we just submit to SRA or something, and then in a future iteration of EORNA, I will get taken up? How does it work?
Yeah, so first of all, you should definitely submit your data. Whether you submit to the SRA or to the ENA doesn't matter, because they mirror each other's data set anyway. We use the ENA because, well, we're based in Europe and we want to support European, but we also it also has the best API for the queries. The NCBI is rather limited. We don't currently have plans or time or funding for an update for the database, and it's a shame that our plans for automation have somewhat been thwarted by the whole problem with the metadata. And really, we had at the beginning of the project the vision that we'd be able to just periodically rerun our pipeline. It would automatically discover more data, quantified, and then we would be able to import it into the database quite easily. But really, because the metadata is in such need of manual curation, this whole scenario has pretty much gone out of the window. And until there is a kind of robust solution to the metadata problem, we won't be able to do updates very frequently or very easily. And at this point, we don't really know how this is going to play out in the future. I'm afraid that's the best answer I can give you just now, and it's probably not very satisfactory.
No, it's fine. Actually, we are also aware of the problem with the metadata.
Yeah.
You mean even metadata that are present in ENA and SRA, so they have been accepted by those guys, but they still have mistakes? Because
Yeah,
normally
I
they
don't
also
want
run
to-
some checks, no?
I don't want to, I don't want to speak badly of anything that happens at the ENA. They do a fantastic job. And I think the problem that there is the whole process of submitting data sets needs to be flexible when it comes to the metadata, because there's such diversity in these experiments that you need to have fairly flexible a flexible schema for the metadata. Let's put it like that. But with the flexibility, of course, also comes the problem of inconsistency, and there is probably not enough enforcement of fields being populated, so people can submit a very minimal set of metadata to the ENA. And I have to put my hand up and say I've done that too. I have submitted probably metadata sets that were less than optimal, and it's an issue. It needs a culture change, but it probably also needs some more enforcement at the point of submission. And even with all of that, there's still the issue of heterogeneity. So you have data sets. Overall, the data sets that get submitted are very heterogeneous in terms of the experimental designs and the germplasm that they cover and so on. So this conflict between flexibility and inconsistency will, I think, be there for a while. And there isn't an easy or instant solution to it.
Yeah, no, I can understand. Thank you. Thank you for answering my question. Thank you.
No problem. Yeah, we've answered a question about the API. So, any more? How many people are left? 41.
Okay. I think.
Well, if there's no more questions, then we can probably draw it to a close.
If you have any questions, please do get in touch with us. We can try and answer it or help find a gene as try and find with David.
If you have a specific query and want a bulk download of a subset of the TPMs for particular samples, we can do that directly from the database side of things. So if you've got a specific thing you need, then just email.
Yeah, we should also say the raw TPM data will be on the website as part of the bulk downloads soon, but we're hanging back with that until we've at least got our paper out as a preprint, just in case. So, but that will be available. It's a large data set. It's nearly 40 gigs of TPM data, but anyone, once we've got it up there, is free to download it and to use it in whatever way they see fit. You could do, a gigantic network analysis on it, for example, if you wanted. We haven't done that yet. There's all sorts of possibilities, but that data will be available and it'll be a very valuable, very large data set. Yeah, so if there's no more questions, then will we just draw to a close?
I think so.
Okay.
Thank you all very much for coming, and it's been a pleasure to present this to you, and hopefully we'll get lots of interesting questions and feedback. If you find any bugs, also please contact Linda, their email contacts on the front page of EORNA, and no doubt between you all, you will be able to find some bugs. There's always some. Thank you and goodbye.